maybe we should get started.
This is actually my second Provost lecture. So I'm trying to do something like a bit different from the previous one. So I think the previous one I talked a lot about maybe the background before joining OIST. So obviously I cannot do that again because otherwise that would be a bit too repetitive. So I will try to talk perhaps a bit more about science and about some of the work we did recently on termite genomes. But yeah, let's me recapitulate still a very little bit how basically we got there. So I arrived at OIST in May 2017 to establish the evolutionary genomics unit. And for my first Provost lecture, there was about 5.5 years, basically, that passed, right? So I had quite a lot of like contents to say. Basically, 5.5 years based on OIST standard is about 11% of a scientific career, assuming that you start at around 20 years old and that your contract is until you are 70 years old, right? So assuming that, you basically have a career of about like 50 years. So 11% of a scientific career is quite a lot of content. So I had quite a lot of things to say at that time, especially that I also talked to what I did before, right? But this time I have only 3 years between the previous Provost lecture, between the series 3 and the series 22, which is only 6% of a scientific career. So I have much less to say. But still, I'll try to make up something that could be of interest for a general audience. Yeah, obviously, I'm not going to talk only about my own work because a lot of the work that is done in my unit is actually primarily done by the people from my unit. So here is one of the latest picture of the member of my unit with some newcomers below. Yeah, so I came to OIST to establish the evolutionary genomic unit, but before coming to OIST, I never really did actually evolutionary genomics. So I did a lot of different things with termites. I was doing like ecology of termites, looking at the composition, the community's composition in different forests, also the diversity, like how basically forest type affect species diversity. I did a lot of taxonomy, so describing new species, new genera. I also worked on termite defense. So for example, on this species that has the worker have the ability to explode in a sense. Basically, their body wall rupture and they mix two compartments together to turn something non-toxic into something toxic, right? That just help them. It's a kind of weird defensive strategy that can evolve when, as a worker, you basically have nothing to lose. You are basically not going to reproduce. So sacrificing your life is just the same as not sacrificing it. You will not reproduce anyway. But this is not obviously evolutionary genomics. So the closest to evolutionary genomics that I did at that time was probably phylogenetic reconstruction. So I did quite a few phylogenetic trees, which is somewhat let's say, the beginning of doing evolutionary genomics. So I use this tree to reconstruct the historical biogeography of termites. So trying to understand basically how, what's the origin of the distribution of modern species. But yeah, so while arriving here, obviously, like I didn't create a unit to basically do what I did, but basically I named it based on what I wanted to do in the future, right? Which was really just like evolutionary genomics. And it took actually quite some time before I could say that now finally I'm doing some of that. So like before the Provost lecture 3, so like perhaps something like 5 years ago, I did my first work, which I think was like really evolutionary genomics. So we looked at bacteria of these cockroaches. I mean, cockroaches all have Blattabacterium inside their fat body. It's a bacteria that provides a lot of nutrients to the cockroaches, and they have relatively small genomes, only 600,000 base pair, which is very small actually for a bacterial genomes. And it's quite interesting to study the evolution of this genome because they all look very similar. All their genes are synthetic. They all have similar contents, similar gene content. They do not acquire anything horizontally from foreign bacteria. So the genome is very conserved. And we looked at a series of those genomes, which basically diverged from an ancestor about like 200 million years ago. We looked at these 67 genomes at that time, and we tried to understand how they lose gene, basically how our genome gets reduced over time. And we built this phylogenetic tree, and you can see those long branches corresponds actually to smaller genomes. So like those genomes may be only like 500 genes, while those genomes here are perhaps like 600 genes, right? So you have like a difference in number of genes, and they are long branches, right? The small genomes are on long branches. So what we found is that there's a strong correlation between mutation rate, basically, and gene loss. So as you accumulate mutation, you lose gene, right? And the faster you do it, the most gene you lose. But selection did not matter too much, right? Like based on DNDS analysis, which look at how selection basically is efficient on those genomes, it did not affect much basically the loss of a gene. So it's primarily something like if you play the tape faster, you lose more genes, basically. And this is not a linear relationship. It's basically an exponential relationship. So it just accelerates over time, right? You first do lose a few genes. It doesn't affect you so much, but as you lose more and more genes, it just accelerates. So this is like the first work that I really did on evolutionary genomics, at least that I can consider as evolutionary genomics, and that was published like perhaps five years or so ago. What we started also at that time is what we call the termite genome project. So the idea was basically to sequence a bunch of termite genomes, a very simple idea, right? And to just look at how these genomes evolve. So a lot of genome projects are basically sequencing one genome, and based on that genome, you try to look at the characteristics of the animals or the plants that you basically sequence, right? That's most of the genome paper, basically. It's a typical genome paper. Like you sequence a genome and you try to link it to the biology of the organism. Like here, the idea was a bit different. The idea was really to sequence a large number of genomes so we can compare them and we can study how they change, really like different compartments, different parts of those genomes. And this is something that was done for a large part at OIST. So you can see the pictures of the main contributor here on the screen. It was also done in collaboration with some German team and team from Czech Republic. So the rationale was basically at that time in 2019 when we started with this project, only 3 genomes of termites were available, and those genomes were not particularly good. So the N50, which basically tells you the contiguity of those genomes. The N50 was relatively small, 2 megabase, meaning that they were very fragmented. All these chromosomes were broken into perhaps like 20 pieces, basically on average. So not that great contiguity. And plus it was only very fragmentary, right? Like you have a really large number of species and only like a few species were basically sequenced. So it's a bit difficult to understand how genomes evolve when you just have a few points, right? So you want to have a lot more points. So so basically the fragmentation, the N50 is about 2 megabase, and the normal chromosome size of a termite genome is perhaps around like 50 megabase, meaning that we should have like an N50 which is much larger, basically, if we wanted to have a chromosome level assemblies. So few termite genomes were available, and those genomes available were actually generally of relatively low quality. So we sequence using the new sequencing facility, partly using the new sequencing facility at that time of OIST. We sequence altogether like 45 termite genomes and 2 cockroach genomes, and we also used Hi-C. So this is a technique that basically scaffolds the genomes. It helps you to patch together some contigs and to really reconstruct basically the entire chromosomes. And we sampled the species to really cover the entire diversity, right? So we had 11 termite families out of the 13 families and 12 subfamilies of Termitidae. So we really have a good representation of the termite tree of life. And here it's just like some general statistics of those genomes. and the L50, so like the number of contigs that make up 90% of your assemblies, is relatively small for perhaps like 20, 25 genomes. So you have an L50, L90 of about 12, 13, 14, 15. That means that all your genome is largely contained into just like a few big scaffolds, right? So you basically have a chromosome level assembly for those genomes. So altogether we had 26 scaffolded genome and 22 were basically chromosome level assemblies. That's of course like all the technical details. And yeah, I decided to make this presentation primarily on this data set because we started actually having some paper published on this topic. So like these two papers were published just at the end of last year, a few months ago, and this one is in press now. And we still have a series of additional papers using these data sets that are now in preprint and that we are trying to publish. So we are going through this series of being rejected from the better journal and going down. I guess this is the normal way how science operates. So I'm going to talk basically about this project, about what can we say basically out of this 45 or so termite genomes, and basically trying to unravel termite genome evolution. So before I really enter into the topic, I'll take advantage of this to really present a little bit my beast, my favorite little beast, and to talk a little bit about what are termites. So Marco already mentioned actually what are termites, so it's a bit of a spoiler, but I'm still going to repeat, right? So termites are not ants, they are not wasps, they are actually indeed some a special lineage of cockroach. So they are some social cockroach, and they have as a sister group this Cryptocercus, which is a subsocial cockroach. So subsocial meaning that the parents take care of the kids. They take care of the babies until they reach adulthood, pretty much like human, basically. And yeah, here you can see a picture of Cryptocercus and Mastotermes. So we always think like termites don't really look like cockroaches, right? I mean, you feel that, yeah, they look relatively different. I mean, they look like this kind of whitish little creature, like the most basic insect you could imagine. But actually, if you think about like this kind of young larvae, here it's a Cryptocercus larvae, they resemble a little bit like a kind of a whitish walker, right? Like something relatively simple. So that's a bit reminiscent of like here Mastotermes, which is the most basal termite species. So termites are social, but they are also feeding on wood. So they may be quite well known for that property. So in Okinawa including, I mean, for people that have a wooden house, for example, they don't really like this, the fact that termites actually like wood so much. And they basically feed on wood in association with a series of microbes. So they also sort of microbes in their gut. They have a lot of bacteria, archaea, and for many species, they also have this very big protist that you can sometimes see with bare eyes, actually. They are so big that you can see them with bare eyes. And they are primarily the one that help digesting the wood, the lignocellulose inside the wood. Termites also produce their own set of enzymes, but yeah, their contribution to wood digestion is very minimal, right? It's primarily the bacteria, the microbes basically, that digest wood. And obviously, if you feed, I mean, lignocellulose is the most abundant biomolecule on Earth. So if you feed on the most abundant biomolecule on Earth, obviously you can be yourself very abundant, right? Because you have a lot of food. And they are extremely abundant. So here, for example, is pictures of savanna in Australia. And you see like all these different mounds. So all mounds basically contain some hundreds of thousands of individuals. So you have like tens of millions of individuals of that particular species in just 1 hectare of land, right? So they are extremely abundant. So much so that they make up about 25% of the animal biomass in tropical rainforest. So actually ants and termites make up together perhaps like half of the animal biomass. So we think about all these big, large animals in the forest. I know like charismatic jaguar or so, but actually what you see if you go to the primary forest is really termites and ants. That's what you see everywhere all the time, right? And because they are so abundant, they also have important function in the ecosystem. So they degrade the wood, but they dig tunnels, a lot of tunnels in the soil, so they improve water infiltration, they provide nutrient to the soil. So they are quite important for this ecosystem, and their activities basically improve plant growth, which is sometimes something contradictory. Like if you kill all termites from a field, you may decrease actually the productivity of that field rather than increasing it. So termites are basically an important group. That's the take-home message, and they deserve more attention. So now let's have a look at these 45 termite genomes. So I'm not going to talk about everything we did with this genome, but just like a few topics which I feel are kind of interesting to report here. So the first topic is about the lifestyle of termites. So they are eusocial, and we were wondering basically what does that change basically when you are eusocial? Like, does that affect your genomes when you switch from a solitary lifestyle to an eusocial lifestyle? So you have like different castes, so you could expect that's maybe an increase in complexity of those genomes. And then the two other topics are about transposon. So I will talk about like how transposon evolved with termites and how we can use them basically to build phylogenetic trees. And I will also talk about the role of transposon in termite evolution. So transposon are generally considered as selfish elements, or at least it's what we have in mind. They just copy themselves randomly in the genome, or less or more randomly in the genomes, but they also can have some function, right? Because every new copy of transposon is a mutation that can have some consequences and can affect the phenotype of the individual carrying this new mutation. And this is a lot the work of Zhong Liu, who is just there. And he's a PhD student in the unit. And yeah, very good PhD student. I did a lot of work. So that basically these three papers. I'm primarily going to talk about these three papers. So, okay, let's start by do genomes of eusocial termites differ from those of solitary cockroaches? So generally, you have like major transition in life. So one of the major transitions is when you move from unicellular organism to multicellular organism. Social insect people like to compare this major transition to the transition from a solitary lifestyle to a eusocial lifestyle. So in a multicellular organism, you have like different cells that specialize in different functions, right? And here, similarly, you have like different individuals that specialize in different castes, which have different function. You know, of course, it's a bit exaggerated, I think, the comparison, but yeah, you know, we need to sell our science, right? So that's how we do it, basically. So termites is a good example for that because cockroaches are solitary. This Cryptocercus is subsocial, so something like between solitary and actually eusocial. And termites here are eusocial, so they have their individuals that are partitioned into castes. And we already knew that there are, of course, some differences between termites and cockroaches. And one difference is that the genome of termites is relatively small in comparison to the genomes of cockroaches. It's still relatively big for an insect, about like 1 gigabase, but cockroach genomes are generally about like 2, 3, 4, 5 gigabase. Sometimes they can even be larger than that. Actually, they can be larger than a human genome. So we looked at the main characteristic of termite genomes, trying to look at what could basically explain their eusociality. So I mean that's like very general characteristic, but does that eusociality basically have some consequences on the general architecture of the genomes? So we looked at, of course, like the genome size, but also some other characteristics like gene number. You could expect that if you have more castes, you have like more different type of individual. Maybe you need more genes for that. Maybe. So we look at all these, but actually, what you can see is that you generally have a reduction, right? Like below here, you have the two cockroaches, so a solitary cockroach, and they have bigger genomes. They have more things in their genome, more exons, they have more genes. They have more complex genomes, in a sense, than termites. While Cryptocercus, here just above Blatta, has a genome that actually looks very much like the one of a termite. But they are not eusocial, right? They are subsocial. So we tested these evolutionary models, trying to think about like how to position Cryptocercus. So the idea here is to kind of define, okay, so this characteristic that we see from termite genomes, are they something which is specific to termites and potentially linked to eusociality? Or is it something actually that occurred before the origin of eusociality, in which case that has nothing to do at all with the fact that termites are eusocial, right? So we tested these two different models. One is you basically get cockroach on one side and Cryptocercus is placed with termites. And the other one is we put Cryptocercus and Blatta together against termites. And we compare these two sets of models. And what we usually find is that for most genome characteristic, you have termites, Cryptocercus really look like termites. So all the small genomes, the number of genes, the reduced number of genes, all these characteristics is things that occur actually before termites became eusocial. So like the evolutionary dynamic of Cryptocercus genome resembles termites rather than cockroaches. So termites and Cryptocercus share similar genomic features, which were required before basically the origin of eusociality. So that's like the graal maybe for people working on social insects. Like they want to look at what is the special thing, what is this special characteristics actually that make this social insect social actually. You must be something because it's such a like big transition in evolution. So far like nobody never really found something really conclusive, right? And when we look at termites, like at least at general architecture of the genomes, it doesn't seem that there is anything special about eusociality, basically. And we looked also, I'm not going to go too much in the details, but we look also at some other characteristics, like for example here, the K-zymes. So the K-zymes are the enzymes that are used to degrade wood, to degrade lignocellulose. They also have some other functions, but it's one of their primary functions to degrade lignocellulose. Obviously, if you feed on wood, you are expected to need this type of enzyme, right? But interestingly, you actually see that you have a reduction of those genes in termites compared to cockroaches, which makes sense because termites primarily rely on their bacteria, on their gut microbes to digest wood, not really on their own enzymes. So the general characteristic of termite genome is really a reduction, like a shrinking both in size, but also in number of genes, including in genes that we would expect potentially to increase in number, like K-zyme that basically would help them potentially to digest wood. So yeah, so basically we didn't really find anything conclusive about what part of the genomes basically change linked with eusociality. The transition of eusociality was basically not accompanied by an expansion of gene number or an expansion of genome complexity. It seems actually the opposite, that it basically was a shrinking in number of genes, shrinking in the size of those genomes. Yeah, so let me now talk about transposon, about how transposon basically evolve with termites. So basically the idea here is really to try to look at how much of a horizontal component and vertical component is there in the transposon landscape of termites. So it may appear like a bit surprising, but actually we know that the landscape of transposon correlates with the phylogeny, but we don't know so much like how much it correlates, right? It's something which is a bit tricky. So we don't know how much is like horizontally acquired and how much is vertically acquired when you look at long evolutionary time scale. Because transposon jump inside the genome, but they can also jump between genomes. I mean, obviously, like any organism, the genome of termites is largely made of transposon. So if you look at any genomes, there's always like a large proportion of the genome is actually some transposon, some of these repetitive elements that jump across the genomes. Because transposon are really genetic elements that can be considered selfish to a certain extent and are able to replicate themselves inside genomes. So a typical cockroach genome is about like 50% of transposon. In the case of termite genome, it's a bit less, but you have like 30%, 40% of transposon basically that make up the genomes. So you obviously have a very strong correlation. So like the correlation is almost perfect between the proportion of transposon that you find in the genomes and the size of that genome. So when you see the fluctuation in size of genomes for many lineages of organisms, generally a large part of that is due to the transposon content. So we try to characterize this transposon landscape, and we annotated all the transposon into families. So like here are the main classes of transposon that you find in termite genomes, which basically you find them also to a certain extent in human, right? So it's general categories of transposon that you find in most eukaryotes. Altogether, we found about 38,000 transposon families that are distributed across these 47 genomes. So that basically constitute our pangenome transposon libraries. Obviously, some species, some genomes have more transposon than others. And yeah, like in this Termitidae, which evolved perhaps like around 60 million years ago, they tend to have more transposon families. So they have larger genomes and they have more transposon families than other termites. So they can have around like perhaps 15,000 transposon families, while some other termite genomes contains a smaller number of transposon families, but it's always in like the multiple, like many thousands basically of transposon families that you find in a single genomes. So yeah, you can plot also this transposon landscape in a different way. Here basically, so we analyze 47 genomes, right? And for every families here, it's in how many genomes we find these families. So you have a lot of 1, which basically corresponds to families of transposon that you find in a single genome. And you have some 47, which basically corresponds to families of transposon that you find across all genomes we analyze. Interestingly, there are some peaks inside the distribution. And these peaks actually corresponds to the number of genomes of relatively big clades of termites we analyzed. So these peaks here corresponds to, this peak in 32 corresponds, it's linked to the fact that we analyze 32 genomes of Neo-Isoptera, which is a big clade of termites. So a lot of basically families of transposon are specific to this Neo-Isoptera, and you don't find them in any other basically group or in any other termite lineages. Here's another peak at 24, and it corresponds to the number of Termitidae species that we analyzed. So a lot of transposon families are really specific to Termitidae. You only find them there, and you don't find them in any other termite lineages. And similarly here for this peak at 8, which corresponds to the number of genomes of Calotermitidae that we analyzed, because a lot of transposon families are really specific to Calotermitidae. So basically what it tells you is that there is really some phylogenetic signal there, right? Like you really have like some families that are specific to some clade. So you can basically use this information to reconstruct the phylogenetic trees. And that would also imply that a lot of the transposon are inherited mostly vertically, right? By inheritance and not really horizontally. So we did that, we built the phylogenetic trees. So here you get a UCE-based phylogenetic tree. So it's a typical phylogenetic tree, which is based on sequence alignment. So you get like sequence, nuclear sequence, and you look at base in this sequence as characters, basically. So it's very standard phylogenetic trees. And here is a tree which is based on the composition in transposon families. And actually, you can see that it looks relatively similar, right? It is roughly the same trees. Actually, these trees based on transposon family composition is better than a tree based on mitochondrial genomes. So the mitochondrial genome is not so big, it's about like 15,000 base pair. And it gives trees that are not as good as this tree based on transposon family composition. So not only you can use the transposon to basically build a pretty reliable phylogenetic tree, but it also basically tells you that the transposon families are primarily acquired vertically, It's not something which is horizontally a transfer, right? It's primarily vertical inheritance, which I think it's not like very surprising in a sense. Actually, it's kind of expected, but it's still something that yeah, it's not yet like, or has not been like really fully demonstrated yet. So yeah, we wanted to go a bit farther because yeah, obviously like you can maybe do better than using transposon family composition. So what we did is to try to look at insertion, specific transposon insertion to build basically this phylogenetic tree. The problem is that yeah, you have thousands of transposon families, and a lot of these families are composed of thousands and thousands of transposon. So it's very difficult to say whether this transposon of that family in that genome is orthologous or homologous to that other transposon in another genome, right? There are so many copies of them. So what we did is to look at elements that we know are orthologous. We know that they are shared by ancestry inside these genomes. So we looked at ultra-conserved elements. So in insects, like termites, these ultra-conserved elements are typically fragments of protein-coding genes. And we know that they are homologous across those genomes. And we look in the flanking regions. We look in the flanking regions of these UCEs, basically assuming that, okay, if this element is orthologous across termites, then the flanking region should be orthologous across termites as well, basically. And we look at the T composition in these flanking regions. So we look at transposon family, presence of transposon family XYZ in these flanking regions with the idea that if we find the same transposon family in the same flanking regions, that basically means that they reflect a single insertion event, basically. And if this is true, we should be able to build a very good, robust phylogenetic tree based on that. So we tested a series of flanking region size from 50 base pair to 4,000 base pair. And as you increase the flanking region size, you expect to have more transposon, but you also expect that the assumption of orthology is going to be less and less realistic, right? Because you have like recombination events taking place, and all this like orthology is going to break down, right? So the rationale here is that transposon found in the same flanking regions are likely the results of the same insertion. So we can use these insertion events to build phylogenetic trees. And yeah, so we did that, and this is the tree that we got, which is basically almost the same as the UC base tree, right? So if you use this kind of approach with this 100 base pair of flanking regions, so like very short flanking regions, you basically get a tree which is almost identical to the UC base tree. So like basically you have a very large amount of phylogenetic information in the transposon landscape. It's just that, yeah, it's a bit more difficult to extract it, basically, and build phylogenetic trees with it. But it's possible. So that's kind of an alternative marker that could be used to build phylogenetic trees as an alternative to sequence-based phylogenetic trees, or sequence alignment-based phylogenetic trees. I mean, obviously, it doesn't need to be an alternative. You can also build them together, right? You can build a tree with both sequence alignment and also this transposon family content. So in conclusion, for that part, our results indicate that the T landscape contains a lot of phylogenetic information, and that this phylogenetic information can be used actually to build phylogenetic trees. So basically, at the time scale of termites evolution, which is about like 150 million years, so at this time scale, most transposon insertion are orthologous across genomes of termites, and you can basically use them to reconstruct phylogenetic trees, pretty accurate phylogenetic trees. So there is some horizontal transfer, but the process is slow, right? So I don't know how far we can go. If we try the entire insect, which is maybe 350 or 400 million years of evolution, I'm not so sure if we will still be able to build good phylogenetic trees. But at least at this kind of time scale, 150 million years, there's a lot of like phylogenetic signals, so largely vertical inheritance. So now let's move to the last part of this talk. So again on transposon, and this time about their role in termite evolution. So transposon are generally they are considered as selfish elements. I mean that's the traditional thinking maybe about transposon, that they are just parasites. They are parasites that copy themselves inside genomes, but that also can jump between between genomes. However, like they are also an important source of mutation. When they just copy themselves inside the genome, they just can affect the phenotypes of the individual carrying this new insertion. And I mean, one very well-known example is the peppered moth that got two phenotypes. So one of them is this white with black dot, which was relatively good in clean environment. And the other one is the completely black peppered moth that was very successful during the Industrial Revolution. There is a transposon in one of the genes called cortex in the black individuals. So basically, like the difference between these two phenotypes is largely a question of this transposon insertion. So insertion of transposon can really change the regulation of genes and therefore create new phenotypes. But new phenotype is what evolution needs to act. When you create new phenotype, selection can basically remove or favor some phenotypes that are less or more successful. So to study basically like how transposon change and evolve inside termite genomes, we reconstructed phylogenetic trees of those transposon. So basically, for every families of transposon, in every genomes, we build a phylogenetic tree. And from there, we can infer some evolutionary distance, right? So for every genomes, we can build a curve that corresponds to the number of transposon that are accumulated over evolutionary time. So we can do that for every single genome. So together we have 47 genomes. We can draw 47 such curves that basically look at the transposon activity, right? So transposon activity depending in a measure of time, which is basically the evolutionary distance. So like here in this curve, we have an exponential descent, which basically suppose that you have a constant rate of transposon activity. But if you have a bump in the distribution, then that indicates that you have period of time when you have increased transposon activity, basically. So this is the curve that we got. When they are not exactly exponential descent, you get like really some bump. Sometimes you get some plateau, sometimes you get really some peaks. And all these peaks corresponds to period of time when you have like increased transposon activity, basically. And you can time it. You can time it in evolutionary distance. So you know that, for example, here you have a peak that may be, I know, like 0.02 in evolutionary distance. So you can really reconstruct the activity of transposon over time using this approach. I mean, obviously, it's not like so informative to have evolutionary distance. What we want is to actually have calendar time, to know basically when actually this event happened. And we can do that by, because we know the mutation rate. Like we can measure based on the genome that we have, the phylogenetic tree that we have, and time-calibrated tree, we know what is the mutation rate for every genome. We can estimate the mutation rate for every genome. So for example, in this case, for Odotermopsis shosteti, which is here, we know that this is number of mutation that you expect per billion year. So it's a very long time distance. But you expect about like, one mutation per billion years. So every site should have one substitution in 1 billion year. So here we know that the peak is about, 0.07. So we know that there was a burst in transposon activity about 70 million years ago, less or more. Of course, there is a certain uncertainties, but yeah, we can estimate that, and we can estimate that for every genomes. So thanks to this independent estimation for every genomes, we can basically look at when across termite transposon activity was more important, basically, when we had some burst in transposon activity. And this is what this plot shows. So like you have the substitution per site here, which basically doesn't tell you so much about the time, and you have actually the time here, which is translated in million years. So you can see that it's not completely random, like you have some peaks of transposon activity. Of course, there is a lot of uncertainties to this analysis, right? So it may have been like an exact timing, but it may have also been like some fluctuation. But you have kind of a peak around 20 million years ago. so somewhere maybe at the boundary of the Oligocene and the Miocene. And you had another peak also about 70 million years ago, so not too far from the Cretaceous tertiary boundary. So you had two different peaks of transposon activity. So the first interesting thing is that these peaks occur in parallel. So across all these genomes, so it's not really just like one genome in particular, but all genomes had this kind of burst of transposon activity, which is roughly 20 million years ago. And obviously, you know that you can see that there is some variation. There's also some imprecision in our measurement, but roughly around 20 million years ago, there were some bursts in transposon activity. And similarly, about 70 million years ago, there was also bursts in transposon activity, basically at the time, roughly around at the end of the Cretaceous, beginning of the Tertiary. So that's quite kind of interesting, right? There must be like something that happened at that time for all termite species, basically, to independently experience this burst of transposon activity. So like here, maybe it's easier to see it on this graph. You can see this peak, basically, that corresponds to period of time when transposon activity is increased. So we can actually know roughly what happened at that time because we have some other work done in the unit where we looked at the diversification rate of termites. So we have these very large phylogenetic trees with thousands of tips, and from there we can infer basically when there was some burst in diversification on the tree. So yeah, you can see it on this big tree. It doesn't tell you so much, but yeah, here is the graph where when you just look at, when you summarize this tree into roughly the diversification rate, you obtain like this burst roughly 70 million years ago, and another one roughly 25 or 30 million years ago, right? So you had some bursts of diversification at that time based on the modern termite diversity, right? When we look at the modern termite diversity, build a tree, we can see that there is an acceleration of the diversification of termite around that period of time. And that's basically match the timing of transposon activity, right? Like if we look at the two, it basically coincides. So you basically had at the same time increased diversification rate, so termite diversified a lot, and at the same time you had this increased transposon activity. So the idea here is that the transposon activity, the increased transposon activity, would basically help with diversification. So when you diversify, you need new phenotypes, you need new things to appear, right? And selection basically would basically act on this. So transposon would really have some adaptative values in this case. I mean, obviously, it's only a coincidence, so you may think that, okay, that has nothing to do with each other. So what we did in addition to that is actually to test it, to test if we have a correlation. And we can do that because we have trees where we estimate the diversification rate. So we can have rate of diversification on every branch on the tree. And we have trees which are based on transposon, right? Which are reconstructed based on transposon insertion. So we can know from this tree, we can also estimate the rate of transposon activity. So like on the same tree, we can estimate two sets of rates, one diversification, and the other one is basically transposon activity. And we can see how much it correlates, basically, which is a bit better than looking at two graphs that looks like the peak is at the same period of time, right? So it's what we did. So here are 4 different analyses. So I mean, they differ by the trees that we used, but they basically say the same. So we have a relatively good correlation. Yeah, we see 0.3, R-square of 0.3, so 30% of the variation is explained here by this diversification and rate of diversification and rate of termite insertion and deletion. You know, obviously, you need also to consider that all these estimation of rates are actually quite imprecise, right? So I mean, having a R-square of 0.3 is, I think, pretty good. So that's basically show that there is some kind of coincidence, right? Like the diversification of termite is somehow linked with transposon activity, and this at geological time scale, at a time scale of like hundreds of millions of years. Okay, this is all I wanted to talk about in terms of science. So let me recap very briefly about maybe the evolution, not this time of termite genome, but of the unit. And yeah, so between so only 3 years of time passed between this Provost lecture series 3 and series 22, and the lab actually changed quite a bit during that time. I think this is something that we all experience. I mean, like when you are postdoc and 2 or 3 years after you go back to your lab, then you realize that half of the people are gone. Sometimes more than half, actually. There is always like a quite high turnover in academia. So yes, there has been a turnover also in my lab. Good thing is not too high, because I think that you don't want the turnover in your lab to be too high, because otherwise there is a problem with you, I guess. So the fact that it's not too high, not too fast, means that, okay, there are at least some happy people in my unit that can stand me for 5 years, maybe. So this is like the pictures of the lab in 2018. Actually, none of the people present here are still in the lab. It completely changed in 9 years. I mean, some of them, like Alej now got an independent position, so that's, I mean, I guess it's good. So somehow it's just like branching off. That's a picture of the lab about 4 years ago. So now a lot of faces are still here. So at a 5 years time scale, we still retain quite a lot of people in my lab, which I think is reasonably good, with some new addition at that time. And here is like a picture of the lab as it is now, basically. So it's changing, but I think it's good. Some people are leaving. The good thing is that they are really successful people in my lab, and I'm very happy about that. I mean, to finish, of course, by a very personal note, I think my kids still like insects. So that was a picture of Laro in 2022. At that time, he was very keen on his museum, pinning some insect, but not only insect, actually anything that he could find, really. But including insect, and you can see like the semi there, and they are still into semi. So that's a more recent pictures. I mean, it's not a good time for semi now because it's winter, but during summer they like to basically go on the tree, pick the semi, and make their collection. It's not really a collection; it's more an accumulation, actually, of semi. But that shows that they still have a certain interest in insect, in other things as well, of course. But at least, I'm maybe doing a reasonably okay job on that side as well. And that's it for this talk. And I want to thank you for your attention.